Okay, chapter 5, coping with environmental variation, and this is related to energy, both we'll learn about autotrophs and heterotrophs. Key concepts here are organisms obtain energy from sunlight, from inorganic chemical compounds, or through the consumption of organic compounds. Radiant and chemical energy captured by autotrophs is converted and stored into carbon-carbon bonds. Environmental constraints have resulted in the evolution of biochemical pathways to improve the efficiency of photosynthesis. And heterotrophs have adapted for acquiring and assimilating energy efficiently from a variety of organic sources. Energy is a basic requirement for all organisms, and without energy inputs, biological functioning ceases. We all need some source of energy. And organisms use many mechanisms to obtain this energy. Energy exists in many forms: radiant energy, sunlight; chemical energy, energy stored in the bonds of food molecules; and kinetic energy, the movement of molecules, typically is measured by temperature. Which is kind of interesting, like temperature is really just kinetic energy. It's just the speed at which molecules are moving around in a substance. Chemical and radiant energy are captured by organisms for growth and maintenance, and kinetic energy influences temperature and rates of chemical reactions, and thus the metabolic rates and energy demands of organisms. So generally what that's saying is our food, our energy is derived primarily from the capturing of chemical and radiant energy. Obviously we don't directly take in radiant energy, but we consume it indirectly via chemical reactions, via the foods we eat. And the kinetic energy influences the way that those reactions work. This should be reviewed, but autotrophs assimilate energy from the sunlight, i.e. photosynthesis, or inorganic compounds, i.e. Chemical synthesis, chemosynthesis, sulfate-reducing bacteria, things in hydrothermal vents, things like that, and convert to chemical energy bonds of organic molecules, primarily simple sugars, and then it's kind of built from there. Heterotrophs obtain energy by consuming organic compounds from other organisms, originally synthesized by autotrophs. Heterotrophs include detritivores, earthworms, fungi, parasites, and herbivores. Consume live hosts, don't necessarily kill them. So a cow can graze on grass and not kill it. And predators capture and consume live prey animals. Some plants are haloparasites. They get energy from other plants, i.e. they are heterotrophs even though they're plants. And dodder is a common haloparasite. Here it says it's an agricultural pest. We have it here. If you go to like the salt marsh over at Cherry Point, you'll see a lot of dodder over that. So we do have it here in natural systems. It's a big issue. Mistletoe's a hemiparasite. It's photosynthetic, but it gets its nutrients in water and some energy from the host plant. So it's sort of in between being a full parasite and somewhat self-sufficient. Hemi mean like half, right? Hemisphere. So it's half a parasite, basically what they're saying. Here's an image of dodder, and you see kind of wraps around plants and can eventually pull them down or shade them out. This is a graph of dodder biomass and the host biomass, showing that as you get more dodder, you can reduce the host biomass. And so there's a negative impact as well, that they're not just like getting a free ride. This is an example from stinging nettle, and it can actually reduce the biomass of that. And then this is mistletoe. It's the green you can see here growing from a tree. Some animals capture and consume photosynthetic organisms or live in close relationship with them. This is called symbiosis. It's an example of a sea slug that consumes algae and then moves the chloroplasts into its skin, and then those chloroplasts are still working. They're still fixing carbon from the sun. Other examples, corals are a good one, where they have zooxanthellae, photosynthetic algae that they host. And some of our local sea anemones do that as well, that they can actually bring in chloroplast and sort of the green sea anemone, and they'll get some amount of energy from the sun and some amount from the food that they eat. Similar to corals as well. Corals still consume animals that come by and stick in their tentacles. Most autotrophs use photosynthesis. Sunlight provides the energy to uptake carbon dioxide and synthesize organic compounds. Although chemosynthesis is important, this is energy from inorganic sources. In both, energy is stored in carbon-carbon bonds of organic compounds. Ecologists often use carbon to measure energy. What that looks like is, if we say this forest fixes so much carbon per year, or certain parts, we measure primary productivity in grams of carbon fixed per year per meter. So that's the way of measuring the amount of energy stored in a system, is how much carbon is it fixing. The earliest autotrophs were probably chemosynthetic bacteria or archaea. The atmosphere is low in carbon dioxide but rich in hydrogen, methane, and carbon dioxide. I think the atmosphere is low in carbon dioxide. I meant to say the atmosphere is low in oxygen. Many of them still use energy from inorganic sources. Again, like hydrothermal vents are kind of a poster child for that. Sulfate-reducing bacteria, which basically use sulfate for respiration instead of oxygen. Chemosynthesis organisms get electrons by oxidizing an inorganic substrate. Electrons are used to generate high energy ATP and NADPH. Energy in ADP and NADPH is used to uptake CO2 and make carbohydrates fixation. So they're breaking the bonds in a chemical, hydrogen sulfide, and then they're using that energy to uptake carbon dioxide and fix it. We're going to go into greater detail about the Calvin-Benson cycle. The book calls it the Calvin cycle, and I have an issue with that. So the most common biochemical pathway to fix carbon dioxide is the Calvin-Benson cycle, catalyzed by several enzymes. It occurs in both chemosynthetic and photosynthetic organisms. I'm calling it the Calvin-Benson cycle because Calvin was the PI of the lab, Berkeley, I believe, in Berkeley. Benson was a researcher working under him. Calvin couldn't figure out how to determine where carbon was being fixed, and Benson came up with an idea that Calvin did support, and Benson went ahead and did it, which is introducing carbon dioxide with a radionuclide, radioactive carbon, effectively. And then he was able to do that, and then kind of basically smash up the chloroplast, run that through a chromatograph. You can imagine having a piece of paper or something more absorbent, putting a marker on the bottom and then dipping it in water, and you know how that kind of like crawls up. And that's a form of chromatograph where compounds are selected based on size. The smaller ones move faster than the slower ones. So Benson was able to do that and figure out what compounds were associated with carbon uptake. He and Benson and Calvin had a falling out. Calvin never acknowledged Benson ever again, even though they wrote the paper together. Calvin goes on to win Nobel Prize, etc., etc. Benson wound up being a faculty at Scripps. He retired in, I think, the 90s, but he was around when I was there. We had a bunch of, retired faculty that would still come in every day. And so a lot of folks have been trying to write that wrong and call it the Calvin-Benson cycle. So I'll refer to it from here on out as the Calvin-Benson cycle. If I make a mistake, I apologize, but I think many botanists have moved to the Calvin-Benson cycle as well. So chemosynthesis, chemosynthesizers include nitrifying bacteria. This is part of the nitrogen cycle, which is how nitrogens are recycled. And so when an animal consumes food and releases that food via defecation and urination, typically those forms of nitrogen are not bioavailable, so they have to go through what's called the nitrogen cycle. And Nitrosomus and Nitrobacter are two main bacteria groups within that convert ammonium to nitrite and then oxidize it to nitrate. Nitrate being the primary form that plants take up. So this is vitally important to recycling that nitrogen and making it bioavailable for plants. Sulfur bacteria occur in volcanic deposits, sulfur hot springs, and acid mine waste. They use hydrogen sulfide and hydrogen sulfide as an energy source, producing elemental sulfur. Elemental sulfur is then used as an electron source, producing sulfate. So these are commonly referred to as either sulfur reducing or sulfate reducing bacteria, because they're reducing hydrogen sulfide or hydrogen sulfate into sulfate. Here's an example from sulfur bacteria thriving in hot springs that have a lot of sulfur in them. This also includes marshes, any place that's has a lot of organic matter and not a lot of oxygen. And so in wetlands, marshes, things like that, there'll be oxygen penetration to the very surface of the sediment, but as you dive deeper, there's no oxygen present. And so typically what they're calling sulfur bacteria only function in the absence of oxygen. And so that the sulfate they reduce and release generates that rotten egg smell. And so if you've been in a muddy marsh and you dig into it or you walk into it and it smells really bad, that's due to your friends, the sulfur bacteria. Photosynthesis produces most of the biologically available energy on Earth. It's also responsible for the largest movements of carbon dioxide between the Earth and the atmosphere. Photosynthetic organisms include some archaea, bacteria, protists, and most algae and plants. Photosynthesis has two major steps: the light-driven reactions. I gotta add Benson in here. Sorry, Benson. Light-driven reactions, light energy is harvested and used to split water and provide electrons that make up ADP and NADPH. Carbon reactions, carbon dioxide is fixed in the Calvin-Benson cycle and carbohydrates are synthesized. The light-driven reactions, light harvesting chlorophyll absorbs red and blue light and reflects green, which is why plants are green. It's because they're absorbing the red and blue light and reflecting the green light, which is what we see. Accessory pigments such as carotenoids help harvest light energy. Sorry, some of these terms I'm terrible at pronunciation, but carotenoids help harvest light energy. The pigments are embedded in the cell membranes or chloroplast membranes. 50 to 300 pigment molecules are grouped into antenna-like arrays. So what this is saying is that we often think of chlorophyll A as the primary absorption pigment for plants or for photosynthesizers, but depending on the spectrum of light that needs to be absorbed, there are different photosynthetic pigments which absorb light at different wavelengths. And so by a plant having multiple types, or by a photosynthesizer having different types, they can absorb different wavelengths. This becomes really important particularly in things like the ocean, where blue light drops out really quickly, and I think reds do as well. I don't know, reds go deeper. Blue light is absorbed pretty quickly. And so if your primary photosynthetic pathways are directed at these lower wavelengths that tend to be more blue, you're not going to have as much success. You're going to capture less light energy. So by having accessory pigments and carotenoids that chloroplasts can absorb light from a wider spectra, which both gives them an advantage. They're able to absorb more light, or allows them to thrive in environments where, say, chlorophyll A might not be the most effective. There might not be a lot of light that chlorophyll A can absorb. Sorry, one more quick thing. Well, we do typically measure chlorophyll A, and so you might have to normalize that by the organism, meaning chlorophyll A values across the spectrum of individuals, or a spectrum of species, doesn't necessarily relate to their photosynthetic capacity. But if you're going to measure within a species, we typically would use chlorophyll A to compare within that species. These pigments absorb energy from discrete units of light called photons. The energy is used to split water and provide electrons. Electrons are released to other molecules on the membranes, where they're used to synthesize ATP and NADPH. Splitting of water generates oxygen gas. The evolution of photosynthesis led to the modern atmosphere of high oxygen levels. This led to the development of the ozone layer and the evolution of aerobic respiration, in which oxygen is the electron acceptor, facilitating great evolutionary changes on Earth. We're going to stop at this slide, and then we'll come back to a few other things. So carbon reactions, the key enzyme in the Calvin-Benson cycle is rubisco, which catalyzes the uptake of carbon dioxide and synthesizes in synthesis of a 3-carbon compound. Rubisco is the most abundant enzyme on Earth. The net reaction for photosynthesis is 6 carbon dioxides plus 6 waters equals a glucose plus 6 oxygens. We're going to go deeper into the different types of photosynthesis, C4, C3, and CAM in the next section.